Thank you very much. So today I'll be talking about opening the DNA past with TrueAllele automation. So let me go back 10 years, almost exactly, to a Washington Post article about Virginia reevaluating DNA evidence in 375 cases. This was an initial example of failed DNA data interpretation. And I'll be talking about what caused that failure and what the response was. But essentially, states started withdrawing their DNA evidence because of changes in DNA mixture interpretation protocols. And that's part of the story as to why DNA hasn't been fully interpreted and why there's scope for going back into the past and taking another look. I know you're all familiar with DNA evidence, but I thought I would put in maybe 2 slides to remind people. And so here's the concept that a person has a genotype. A genotype is the genetic type. It could be all 3 billion DNA letters that you have, 1 copy from each parent. Usually it's just sampled from 15 or 20 different short locations called loci on the human genome from different chromosomes. So this is showing that a person has chromosomes. They're in cells and in the body, and there are a dozen or 2 locations, 1 locus, many loci, that scientists test, and that's indicated with the red bar on the chromosome, and that a person will inherit 1 variant called an allele at a locus from a mother, and another allele from their father, and that pair of alleles at that locus, here it's a 15 and a 16, is called a genotype. The genotypes that are used are short tandem repeats. I'm not going to get into that technology, but they're based on lengths. So I'm showing that the DNA molecule of 16 as an allele variant is longer than the 15. When you have 2 people, there are 2 genotypes. And here we introduce a second person. We sample that same 1 location. I'm showing 1 of the, say, 20 locations that are tested. And we see a second allele pair from a second individual at this location. And maybe that's a 17 and an 18. Those are the names or lengths of the alleles of the genetic variants. I think that's it for the biology. And for the rest, we'll be talking about what happens with data interpretation. So this is DNA mixture data. And this is what it looks like. A laboratory has collected a sample. They extracted the DNA, amplified it with polymerase chain reaction, PCR, making billions of detectable labeled copies. It's introduced some random variation that has to be accounted for. And this is the readout on a DNA sequencer, or also called a genetic analyzer, and this is what 1 of the 20 locations look like. 1 locus. You see on the horizontal x-axis different sizes. That's the length of the molecule, like 138, 153, how many DNA letters there are. The numbers in the boxes are how many repeated units are. That's the length measurement that the scientists care about. Are there 15, 16 repeats, or there 17 or 18 repeats? And then the vertical axis is RFU, relative fluorescent units. That's the amount of signal that's being measured from the amplified alleles, the different genetic variants. And this is what the data looks like. It's a bunch of signals that have been collected from the amplified alleles. Peak heights, whether it's 300 high at 15 or 600 high at 16, is showing us how much has been amplified of each allele. So that peak height information is very important if you're looking to see how much of each person's present to analyze the data. There's a pattern. There are 2 taller peaks from the 15 and 16 from the first person, and 2 lower peaks at the 17 and 18 from the second person. There's also variation in the pattern, like between the 15 and the 16. They're not the same height. All of that is really useful information in fully analyzing data. And without all that information, even the baseline noise at the very bottom near 0, you can't get a complete analysis. Human review simplifies the data. The problem is too complex. It's like a 100-dimensional probability math problem. It's not what people do. And so in order to deal with all this data, most of it's thrown out. That's been the standard methods used in labs for over 20 years. Arbitrary thresholds are applied that cut through the data. I show one in red here. And what that means is that peaks that are above the threshold, like the 15 and the 16, are brought back down to the threshold level. Peak height information is discarded. Under the threshold, those two smaller peaks, which might correspond to what you're really interested in, they're gone. They're treated as if they're zero. So all of this highly informative, rich information, evidence data, most of it's discarded, and you're left with 15s there and 16s there. That's it. That's what the scientists get to see with these simpler methods. So early on, people knew there were problems with these methods. And the main method that's used is called a probability of inclusion. We'll talk about that. And when you have multiple locations and the numbers are multiplied together, that's a combined probability of inclusion, or CPI. You'll be hearing those letters a lot. Most DNA has been analyzed in the US with mixtures using CPI. The US government got involved. NIST is in the Department of Commerce, the National Institute of Standards and Technology. They don't have much expertise in modern interpretation of DNA evidence, but they did collect information about how well labs performed. And they did a lab study 15 years ago, which they never actually published scientifically about what their results and conclusions were. But what they found was that on the same simple two-person mixture, most labs found the data was inconclusive, and when labs did report a statistic, it ranged from tens of thousands, which is 4 zeros after the 1, to hundreds of trillions, which is 14 zeros after the 1. I'll be using the number of zeros after the 1, like 4 is 10 to the 4th, or 1 followed by 4 zeros, to represent tens of thousands. It's much simpler to talk about. We discuss it with juries, and it's standard in science. And we also can see all the results on one scale. There was also bias that people were noting in reports and in published papers, cases where, depending on what the analyst thought the answer was supposed to be when they made the comparison, it would change what the results were. So in fact, whereas you might think that, well, you just put the data into a machine and it calculates a statistic, that isn't what happens. The data are chosen. Comparison is made with an answer of a possible suspect. A human analyst decides which data will be used or not. Only after a person has curated that data in the context of what might be a desired answer is any statistic calculated. There were problems with false positives, and NIST and the FBI suggested adopting a second, higher threshold to get rid of false positives. This was called a stochastic threshold. Stochastic just means random. So the idea was the way to deal with randomness, instead of using a computer to model it with human review, you just keep throwing out more data. So if all the peaks at the first threshold that's lower don't hit the threshold that's higher, you throw out the data. So most human analysis of forensic data is about how much can I throw out? How much can I cut off? When can I not use it? Unfortunately, this did not solve the problem of false positives, as NIST discovered in a study in 2013. Looking at this, at this point in the study, there were 100 labs responding. 70 of them falsely included somebody who wasn't there and provided CPI mixture statistics for DNA. Of course, they were wrong because the person wasn't there. Only 6 out of the 100 groups actually excluded the person who wasn't there, including a South Carolina lab that used TrueAllele. I should stress these CPI methods have nothing resembling scientific validation papers. They're not peer-reviewed. There's never been studied. They're just rules that people follow. I actually looked at data from a lab and analyzed it and published a paper 5 years ago based on data that show that the CPI method is nothing more than an analyst counting up how many locations they subjectively think a defendant is included in. That's all the method is. In fact, the title of the paper is Inclusion Probability for DNA Mixtures is a subjective, one-sided match statistic unrelated to identification information. Essentially no correlation. So it didn't stop people from using it, but there's no scientific basis for it. But the problems didn't go away. Labs continued to use these unvalidated, unreliable methods, and they started shutting down. Prosecutors were concerned that match statistics were changing as the interpretation changed from one threshold to two threshold. The DC lab shut down in 2015. Texas lab was concerned about mixed DNA evidence, and 25,000 lab tests were affected and reanalyzed using yet another version of CPI. So it was kind of a useless reanalysis. So human mixture interpretation is problematic. It's incomplete, discards most of the data, applies thresholds. It's inaccurate. It disagrees with the true information that's present in the data that can be measured. It's subjective. The workflow introduces human bias and exposes the analysis to what a desired answer might be. It's inoperative. There are hundreds of thousands of cases where informative data has just been reported as there's nothing here. It's inconclusive. It's opaque. People reviewing the cases don't know why these choices were made to use, say, only 7 out of 15 locations. And it's biased in yet another way. It can only include people. It can't give a statistical answer that would exclude people. So you end up with mainly inconclusive results from very valuable DNA evidence. So enter TrueAllele. TrueAllele is a method that I invented over 20 years ago. It's a computer solution. It's currently used by Department of Forensic Sciences in Virginia and in other states. The method's complete in the sense that it uses all the data. It doesn't have thresholds. It also considers, by statistical sampling, essentially all possible explanations of the data. It's accurate. There have been 42 validation studies written up, 8 of them published in peer-reviewed journals. It's objective in the sense that the workflow removes human bias. The computer has no concept of what you're looking for. It first solves the problem, producing genotypes from the data, and only afterwards can make a comparison to an individual, whether it's 1, 10, or 1 million people. It's generally accepted. We've sent out reports in 45 states. We worked on the World Trade Center. There are 10 US labs using it. It's very transparent. We provide the math, starting with publications on our patent disclosure 20 years ago. We give the software to whoever the opponent is if we're involved in a case, and we always let the other side test our software at no cost, because the only thing that matters in science is testing. That's the basis of what scientists do in all standards in forensics. It's also neutral. It can statistically include or exclude, or sometimes say there's nothing there. So it gives informative results. That's a synopsis of the difference of what TrueAllele is. What does it do that's so different from human review? Well, TrueAllele explains the data. It considers all the different genotype combinations of different individuals. It doesn't see the suspect. Here is the same data we saw before, and here's one of maybe 100,000 explanations that TrueAllele will try, just considering how to explain the data. So on the taller peaks, 15 and 16, we see 2 blue bars. They represent a major contributor, more than half of the DNA. That might be a proposal with a 15-16 genotype as an allele pair from one person. And then as the genotype at this location from the second person, the computer might propose it's a 17-18, and that's shown in red with a quantity that's much lower. And that pattern that you see of 2 tall blue bars from one person and 2 small red bars from a second person explains the data well. Most patterns in different genotype combinations do not explain the data well. If you propose a 12 and a 13 for a person, there's no support in the data. But the computer's going to try it, and it's going to tell you there's very low probability for many possibilities, but there's much higher probability for these genotypes, one person having a 15-16, the other one a 17-18. That's why it's called probabilistic genotyping. Instead of trying to come up with a one answer that's either wrong or give no answer at all, sometimes it's right. Instead, what the computer does, it says, well, consider all possibilities. In science, once you consider more than one possibility and you assess it, you need to use probability in order to give a description of what you found. TrueAllele unmixes mixtures. In a case like this, it might find, as it separates the mixture data into the different contributor components, the major that's 75% and the minor that's 25%, and works out their genotypes. Here's another picture from the TrueAllele interface that shows that separation into the different components. That's all worked out from the data. We don't tell any of this to the computer. It does it all by itself. One important fact about TrueAllele that has been confirmed in study after study, this one was first published in 2009, the information gap article published in PLOS ONE, is that the TrueAllele results, or in general probabilistic genotyping results, if the models are good, are predictable. There's a straight line. We know that that as you increase the amount of DNA for a contributor, on average, the amount of information, the match statistic, will increase. This chart shows how you go from 0 on the bottom left, 0 DNA input on the horizontal x-axis, and essentially 0 information as the match statistic, the likelihood ratio, or working in these powers of 10, where you see on the scale 1,000, million, billion correspond to 3, 6, and 9 as we add up. That on this powers of 10 log-log comparison, as we increase the amount of DNA, we get up to about halfway, 100 or 200 picograms. It then plateaus off. There's no more information. So it's predictable in the sense that it follows basic rules. More DNA in a contributor, on average, more match information, higher match statistics. So I know this is the part of the talk you've all been waiting for. What does this have to do with Daubert or Spencer in Virginia? The reliability prongs. So let me discuss them briefly with you because I can cover a lot of ground of what's important to lawyers with this. The Daubert prongs are, is testing possible? Has a method been tested? The idea is that under Rule 702 on reliable evidence, for an expert to testify, there should be a method that's reliable, there should be sufficient data, the method should be reliably applied to the data. But how do we know it's reliable? Enter the Daubert prongs. The first prong is testing. The second is error rates and standards for the use of the method. The third is, has there been peer review and publication? Peer review means independent assessment by other often competing or adversarial scientists of a scientist study where scientists have all kinds of bias. They get grant funding, they may get paid, they may have prestige involved. So what peer review has done for centuries is to let your competitors look at your work and deem it worthy for publication or not. That's why in science the method of scrutinization, which the Supreme Court in the US has adopted, is peer review, and also general acceptance in the relevant scientific community. So let's quickly go through those. TrueAllele testing. TrueAllele can be tested, and it has been tested. in that first initial potential disaster with 375 cases that were handed back as reports by the lab back to prosecutors 10 years ago, Virginia had Cybergenetics review 144 of those cases, and on the 72 that went to court, we confirmed the results with additional TrueAllele processing, and then issued reports, and we and the lab testified on at least a dozen of them. And so based on that testing that was done, we actually had the basis for an extensive study where we could compare human review with several methods of CPI with TrueAllele. I'm not going into all of that today. That's in lots of other talks. That's not what you asked me to talk about today, but it's interesting. The main axes of validation that we look at that are in standards are described here. Is the method sensitive? That means if someone did leave their DNA, is TrueAllele picking it up? We're getting a match statistic that's inclusionary. Is the method specific? If somebody didn't leave their DNA, are we giving an exclusionary result? And if not, what's the false positive rate? This is great for producing error rates. Under Daubert, error rates can be high or low, but by looking at specificity, we can produce them. And to what extent is the method reproducible? To solve these probability equations, what's needed are randomized methods that don't give the identical answer each time. They sample. Kind of like in an election, where if you do polls, you will not get the same numbers twice on successive weeks. You'll get numbers that are close. In fact, if you do it on the same day, they're not going to be identical either. But are the numbers close? Is your pollster reliable? Same thing's true with these methods. And so by validating them and testing, running the method twice, statistically looking at the differences, we can show that the results are within a factor of 10. It's as match statistics. The results are reproducible. I did want to comment on one thing we noticed in the paper because it gives you a sense of comparing computer and human, and that's looking at the error rates. Error rates had not been computed for human review in a major way before. But in this study with Virginia, we had an opportunity to do that. We go into this when we do admissibility hearings in a lot of detail. So first, with the specificity, the notion of error rates, we can look at rare events where we know that people have not left their DNA and ask what the error rate is. In summary, I won't go into detail, for match statistics that are also called likelihood ratios or LRs that are over 100, a log value of 2. So there's 2 zeros and 100. The error rate in this study on this data was about 1 in a million, or 0.0001%. Great, there's an error rate. What about CPI, the method from 20 years ago, with an analytical threshold which the state used for 10 years? Well, their analysis found 5 false positives in 81 of the 100 comparisons that we studied. They agreed the computer was right. That gives us an error rate of 5 in 81, or about 6%. So that's an error rate. So when a result is reported, you're getting 6% false positives. Now, this modified CPI with stochastic thresholds was supposed to eliminate it. It also eliminated most of the information. They could only make 53 comparisons instead of 81 because they threw out so much data they couldn't give results. And on average, the match statistics dropped from millions to hundreds. By throwing out 2/3 of the data with stochastic thresholds, that threw out 2/3 of the zeros. 6 zeros is a million, and 1 followed by 2 zeros is 100. They threw out 4 zeros. And so you would hope that now we're finished with false positives. Nope, still have one. So there's a 2% error rate. So for throwing out all that evidence and all that information, it didn't solve the problem. TrueAllele's been peer-reviewed. Of the 40-plus validation studies, 8 have been published in peer-reviewed journals. The studies involve both mixtures synthesized in a lab of known composition, those are the 5 on top, as well as validation studies that come from actual casework, where we're looking at what comes in from the field. Those are the three on the bottom. Both are really useful in establishing what the system can do. Virginia wrote its own validation paper without us, that's the fourth one down, and published it in the Journal of Forensic Sciences, in addition to the one they published with us. That's the last one on the page. I do want to note one of these papers gives more concern about human review. This was a study we published with New York State 8 years ago. We had published another study that had asked, when a human review gives an answer, how well does the computer do? But this asked the other question. When the computer gets an answer, that's indicated by that blue cloud in the background, how often does human review give an answer? Well, it generally gives a lot less information, but this was the first study that showed that 70% of the time on simple DNA mixtures, human review failed to give any answer at all, which surprised us, but there was basically no reporting on simple 2- and 3-person mixtures, 70% of the time. Those were 85 cases that we looked at. Next, for general acceptance of TrueAllele, this just gives a summary. The math and the algorithms are 25 years old. We developed the computer system starting 20 years ago. It's used by 10 labs in the US. There have been over 40 studies. We've given probably hundreds of talks at this point, but at least 100 talks. We've certified hundreds of analysts. There have been 29 admissibility rulings, Spencer, Fry, Daubert, all sorts of standards. Had one yesterday in federal court down here in New Orleans. Fortunately, we haven't lost one yet in the US. That could happen. It depends on the court, whatever's going on. But it's been considered to be reliable for over 10 years across 14 states and federal jurisdictions. We have gone to trial with it over 100 times, and the crime labs who use TrueAllele have submitted tens of thousands of reports. With a lot of education, we do many CLEs. I've done them in Virginia for your organization. And we work on making the ideas understandable, and we've issued some type of report in 90% of the states. It's generally accepted. There's no scientific controversy about the method. No one has done a study that says this is all rubbish in any peer-reviewed way. It just hasn't happened. Here's a listing of the 29 admissibility rulings, the latest ones on the bottom. And Virginia is towards the top. That was one of the first admissibility hearings in Colonial Heights, Virginia versus Matthew Brady. Judge Sherrod admitted it in 2013. So now, this is the part that should be more fun for you about cases and what are the implications. I'm going to go into the past in just a second, but first, let me show you the difference that TrueAllele can make. This was a case, I think maybe it was in Arlington. It was in the northeast of Virginia. And this was a man, David Black, who was accused of murdering his wife in the bedroom. And this, what I want to show is what you get out of real information. On the left, we have a list of 3 evidence items. A hat, a master bedroom light, master bathroom light switch. And on the right, we have the suspect, the decedent, different relatives. The initial analysis is, well, we can't pull apart the relatives, we don't know what to say, and so on. But TrueAllele can pull apart the relatives. It can give answers where it's blank. That means we're not asked to give a statistic. And in addition to being able to separate out what's going on with relatives, by looking at all of these results, and this is typical with larger crime scenes, what we can see is that both the accused and the decedent left their DNA on the master bedroom light switch. But David Black did not leave his DNA on the bathroom light switch, which was argued that he wasn't regularly there, he wasn't supposed to be there. But why is his DNA on a light switch that it's not supposed to be on if he says he didn't enter the house? And so this is how DNA is used when all of these match statistics that are often not reported, that are in millions and so, are not visible to prosecutors or defenders or criminal justice. This was a case that was interesting in Pennsylvania, where there was a car with 4 people in it. The question is, who is holding on to the gun? The lab couldn't say anything about it. They can't unmix 4-person mixtures, but TrueAllele can. And so we were able to provide a match statistic, about 400,000, to one of the defendants. Imagine you're starting off with multiple defendants and you just don't know. There's 4 contributors on the gun. There is no DNA evidence in the case. Everybody's a suspect. After the computers analyzed it, one person is included. He pleads guilty. And now we're getting to the questions, which is, person A was excluded. Charges were dropped because we had more information from the DNA. And that's what the DA's office chose to do. Let me tell you about some historical cases. This was the first exoneration we ever worked on. So far, TrueAllele has helped exonerate or free from prison at least 10 innocent men. This is Indiana versus Darryl Pinkins and Roosevelt Glenn. Many articles, TV shows, I've written about it. I can send you materials on it. 1989, 5 men raped an Indiana woman on a highway in a series of bump and rape crimes that were bump and rob, bump and rape that were escalating. Darryl Pinkins and Roosevelt Glenn and another man were misidentified based on clothing that had been stolen from their car. They were wrongfully convicted in 1991. Darryl was sentenced to 65 years in prison for a crime he did not commit. There was DNA mixture evidence obtained in 2001, and conventional human review found the obvious 2 major contributors, who are of course not the accused, but the post-conviction court said they wanted all 5 because there were 5 men involved in the crime. And so the post-conviction relief was denied, and Darryl stayed in prison for another 14 years. We got the data from the Wrongful Conviction Center in Indianapolis, and with the help of Professor Fran Watson, who super bright, former public defender, really thought about the evidence and helped us ask the right questions. These 5 things were not being done by other, even computer programs back then, and they certainly can't be done by human review. We could compare evidence with evidence, not just references. Secondly, we could calculate exclusionary match statistics. We could reveal a 5% minor mixture contributor. We could jointly analyze mixture data across multiple items to get more information from the same data. We could also show that 3 of the perpetrators were brothers, which these meant none of the accused people were. So we found the 5 unidentified genotypes using TrueAllele, and the defendants were not linked to the crime, and the DA at that point canceled the hearing, dropped the charges, and Darryl Pinkins was exonerated in 2016. So that's a pretty dramatic example of what a computer can do that human review and what's been done in the past throughout the world has never happened. It just doesn't happen. This is a case of Georgia versus Johnny Lee Gates. In 1977, he admitted to a murder, was convicted, and sentenced to death. The case gained national notoriety because of some issues. He was intellectually challenged. He had some mental deficiency. He was brought back to the crime scene for a confession where he touched items from which fingerprints were lifted after he went back. And the prosecutor involved had a history of striking all the black jurors in several capital cases. As the case moved along, some interns from the Georgia Innocence Project discovered 2 new ligatures that seemed to be very involved in the crime, in binding the victim and in killing her. These were 4 contributor mixtures. And the lab's human review, today, by the way, the Georgia Bureau of Investigation uses TrueAllele. but at the time they didn't, and their human review found the mixtures to be inconclusive, which was typical for what people were able to do, say 3 to 5 years ago. We received the data in TrueAllele. I ran it through TrueAllele, and what we found were strong exclusionary statistics for some of the items in the trillions, but a number that's over 1, like 1,000,000 or 1,000, that's an inclusionary number. The bigger the number, the stronger the match strength. Numbers that are less than 1, like 1 in 1,000,000 or 1 in 1,000 or 1 in 1,000,000,000, those are exclusionary numbers. So he wasn't in this evidence. And at the hearing, it was pretty dramatic how hard the prosecutors were working to not acquit this man. There was an amazing theory that was brought forward of, well, there may be 4 contributors on these items of unknown people, so there's DNA there, but Johnny Lee Gates's DNA must have fallen off with the passage of time. I'll tell you that's laughable, but it wasn't explained with humor, it was explained with analogies to the judge that DNA doesn't fall off. But the judge was persuaded, and he was granted a new trial, and ultimately he took an Alford plea and was released from prison after, quite some time. Texas versus Slidell Grant is a case that I got involved in 2 years ago. In 2011, the Houston lab, as expected, failed to interpret a DNA mixture, evidence from fingernails of a 2010 murder victim. And again, it's expected because that's what labs were doing, that they weren't expected to be able to interpret their mixture evidence properly. And the crime lab analysts testify that the DNA was inconclusive. Slidell Grant was convicted of murder and received a life sentence. 2 years ago, the Innocence Project of Texas sent the DNA data to Cybergenetics. And the match statistics that we found, this is represented as the number of zeros. I'll translate. 18 means a billion billion. -12 means 1 in a trillion. So the victim's DNA was connected to himself, but Slidell Grant statistically was not there. The match statistic was 1 in a trillion. What was testified to as inconclusive was just, very strong evidence that he wasn't there. This comes from the TrueAllele report. So in addition to excluding Slidell Grant statistically, with a 1 in a trillion statistic, we also stated in our report that, as often happens, we found other genotypes from unknown people. They didn't match anybody we were given as a reference. This is language from the report. TrueAllele also inferred a non-matching evidence genotype from the fingernails, and the genotype of this unknown contributor has an expected match statistic of 18 trillion. TrueAllele can calculate how much information is in the data without ever making a comparison. We actually calculate the full distribution of probability distribution of what the match statistics would be to the right person and a second distribution to people who aren't there. That's very helpful for error rates, by the way. And we do it in all of our reporting. So we knew that there's somebody there with a very strong match statistic if we knew who the person was. So the question was, how are you going to search it in CODIS? We developed an allele list that could be searched in CODIS, and TrueAllele can make a comparison to the original genotype that we have in order to determine whether or not somebody's there and get an actual match statistic. So we did an unprecedented CODIS search. The lab that uses TrueAllele in South Carolina researched the CODIS rules for a year, developed protocols, and he did a CODIS search out in South Carolina. And we found the killer. He was in Georgia, and when he was confronted, he confessed to the crime. And wow, were we in trouble. I mean, Texas wasn't pleased, the DA wasn't pleased, the police weren't pleased, the FBI wasn't pleased. I would have thought people were pleased. We try to follow all the rules, and we find that an innocent person who's in jail, that the DNA actually comes back to the killer who then confesses. that happened afterwards once the police got involved from Houston and took a look. So ultimately, it took months, despite all this evidence. Grant was released, and a few months ago he was finally exonerated. And this shows the power of when you're using TrueAllele results with your crime lab, like you may well be able to do in Virginia since DFS uses TrueAllele. You can be doing searches to say, oh, I have new DNA evidence. Can I go back and solve the crime? So on a national level, not so much, but possibly in Virginia. This is a case I testified in just before the pandemic in New York. What's notable about it is that the crime laboratory reported inconclusive results. And I'm sure this never happens in Virginia, but it happens in New York. We've seen it several times, and it's dramatic. Seeing the chain of logic that a prosecutor can give as they go from inconclusive results, which scientifically means there's no information, then saying, well, the person can't be excluded, and then in closing arguments saying, therefore they are included, and therefore they're guilty. And I wouldn't have believed it if I haven't lived through it several times when I testify for defendants. So inconclusives can be very dangerous because they can be twisted unscientifically as proof of guilt, when basically 0 means 0. It doesn't prove anything. TrueAllele reanalyzed the data, and the match statistic was 1 in a trillion. After we did our analysis, the crime laboratory revisited their analysis and reported a weak exclusion of about 1 in 1,000. I was also asked to touch on modern cases, so I'll do that now. This is a case in Pennsylvania. The prosecution asked us to analyze it. They couldn't use it. They turned it over to the defense. The defense did use it. And what happened is they saw a lot more match statistics here than the crime lab had seen, particularly these lower numbers, like 1 in a million or 1,000. And what this let the defense do was to say this wasn't first-degree murder. There's signs of a struggle. Josh Huber, who's the defendant at the right column, his DNA is under the fingernails of the 2 victims. This, which suggests that he didn't just invite them over and shoot them, but there actually was a struggle. His blood is on the wall without them. So in the end, Huber was actually acquitted based on the DNA evidence that we provided the prosecution, because a more microscopic view was able to get more results. This case of California versus Manuel Lopez, I think, is one of the most important cases I've ever seen. I gave a talk on it about bias and what happened in this case at the American Academy meeting. I can send you the link if you want to see it. I'm just going to give you a survey of it. It's basically the story of a man in his 20s accused of capital murder. He's accused of rape and murder of his girlfriend's toddler son. Horrible crime. He's facing the death penalty or life in prison. The child's 2 years old, 2 years and 10 months old. Bruises on face, genitals, rectum. Autopsy shows brain swelling, skull fracture, cheek bruises, asphyxia. A rectal swab from the boy showed semen. And now, here's the kicker. The swab matched the defendant's DNA. Okay, so he just goes to the electric chair, right? And we're finished. But that's not what the evidence really showed. There were 75 evidence items that the lab and Cybergenetics looked at. Incidentally, we worked for the prosecution the year before on a different case. So in the county, in Santa Clara, there wasn't a question about TrueAllele's reliability. It's what's the DNA showing. This is what the lab found, comparing 25 of those items against 6 references: the defendant, the mother, the brother, the victim. And here are also 6 items that of unknown people, one of which the lab found, and the other 5 TrueAllele found. And what you see is it looks like there's the defendant and the victim, open and closed. But this is with primitive old manual methods. TrueAllele opens this up. It's showing match statistics to all these unknown people. It's showing match statistics to relatives. It's showing more of the defendant. It's just showing results that weren't there before, and that changes the case. Who are these other people? Why are some of them on more of the probative items than the defendant? Where's the mother? How could the main caretaker not be on 75 evidence items? So we were left with these puzzles that honestly we didn't figure out until shortly before we gave our evidence. One is, where's mom's DNA? Lots of people, where's the primary caretaker? And then there's the rectal DNA conflict. Why was the DNA found from the defendant initially at the hospital, but not later at the autopsy? But a more detailed analysis that revealed the information actually could answer the questions. The final verdict was this: the prosecution was target driven. That's the point of the talk that I gave. The defense was non-target driven. We educated the jury for days at length, and the scenarios that we described much better explained the evidence, and the jury acquitted the defendant of all charges, and the county has given up the death penalty. So this had major impact in California. So let me sum up now on the last few slides. Human review has failed. Science has shown for 20 years that it's inaccurate, unvalidated, it's biased, it gives unconclusive results, it's uninformative, it's not reliable, it's often not relevant, and if you really held a Daubert hearing on it today, I don't know if it would pass. And in the center, this is a quote from a peer-reviewed paper, the journal, the JPI paper I mentioned. Inclusion probability for DNA mixtures is a subjective, one-sided match statistic unrelated to identification information. That's the title. So what's the problem? There have been hundreds of thousands of past cases that have informative DNA evidence, but either meaningless DNA statistics, or most often no DNA statistics, and certainly no exclusionary statistics that might help exculpate people. And that's just a fact. That's the situation across the country. On the other hand, TrueAllele technology, whether it's our product or others, the TrueAllele works, and the automation that we have succeeds as well, which I'll comment on. TrueAllele has been proven in peer-reviewed scientific articles and study after study to be accurate, validated, it's unbiased, it gives statistics. It also gives error rates, which is important. I'll comment on that in a second. It's highly informative, it's reliable, relevant, and it passes the Daubert prongs. We had our 29th hearing in federal court yesterday, and the judge ruled it in. The question was about very low-level match statistics. And based on the evidence, yes, low match statistics are science. The issues go to the weight, not the admissibility. And quoting from the Virginia paper, published in PLOS ONE, that journal, TrueAllele mixture interpretation is a reliable method. It's objective, sensitive, specific, reproducible, accurate, and it's also more reliable than human review. But this is very important at the bottom. TrueAllele is also automated, unlike anything else I know of out there. We built it to be automated. One of the first test cases was the World Trade Center project, where we were able to process 100,000 DNA data files for 18,000 victim remains and process it in a short period of time. It's automated. We can run it on large batches of cases without having people scrutinize the data or make any choices or decisions. I don't know of anything else like that. It's automated and accurate, which leads me to the conclusion: how to open the past. Commonwealth's attorneys are the chief law enforcement officers in their counties. They can request electronic DNA data from the state lab. If you get it, you can send the data to Cybergenetics for TrueAllele processing. And then after processing, your county can receive back, or groups you have, or conviction integrity groups can get back accurate identification information. Not to put a fine point on it, but even groups that use TrueAllele in crime labs, many of them continue to have arbitrary cutoffs or limits, whether they're for or against defendants. They'll limit the number of contributors. They'll limit how much DNA they'll look at. What labs often do is they limit the match results. So really probative results where the match statistics are under, 10,000 or 1 million, some labs won't report. But because TrueAllele can provide, and we do provide, exact error rates on every match statistic, we provide a frequency context for every match number we produce, and therefore we can report everything. If the frequency number, if the error rate is 10%, that's clear to the jury that this isn't great evidence. If the error rate is 0.001%, it's communicating that to the jury. In science, scientists report everything with error rates, and TrueAllele makes that possible. So all these low-level results, as we see in case after case, we actually did a survey of it, and dozens and dozens, I don't know if it's 100 cases, but that we've reported on, very, very low match statistics. But with error rates, that's fine. It's up to the trier of fact to establish the weight of the evidence. And in conclusion, better science leads to better justice, and it would be great to open up the past and make all of this underinterpreted DNA available to modern technology. Thank you very much.